I'm excited to introduce Julia Brown. She's a colleague of mine, friend of mine at Biggle, where she's a research scientist, a viral ecologist. She's going to talk to us today about working with some ocean omics data. But Julia is really the person that introduced me to Python and to the world of Jupyter and got me going in this world. And she helps me all the time to navigate our computing cluster at Biggle. So she's helped me a lot on my journey in this. So I'm excited that she's here to give us a walkthrough on some of her work today. Julia, we are going to be keeping an eye on the Slack and answering the kind of the questions that we can, but if anything comes up that we think needs your attention or that it would be good for you to answer, I'll pop up and verbalize those questions. So you don't need to worry about keeping an eye on that.
Great, cool. Thanks, Kath, for the intro. It's really fun playing with Python with you at Bigelow. One thing that we've learned as sort of friends and coders and scientists is that like we work in very different realms of ocean science. Like I am interested in viruses, which are like super small and like you can really only look at them in like a really small sample relative to the way that Kath looks at the ocean. But we have such a common language in the way that we interact with data because we're working in notebooks, we're working on Python, we're interacting with servers that we have a lot of fun together anyway. And so I'm thankful that you've invited me here. So one question, is anybody doing any bioinformatics at OceanHackWeek this year? I'm just curious.
There are, I'm not sure if anyone's doing any projects directly with bioinformatics, but there are definitely a couple of people that are doing bioinformatics
cool.
in their research realm.
All right, just curious, like who I'm speaking with here. That's great. And so today I'm going to talk a little bit about playing with metagenomics data in the context of ocean science, I guess. But really, I'm just going to showcase, I'm going to introduce sort of like the data, where it comes from, and one particular tool to sort of profile and learn about ocean samples through metagenomics data. So I'll start with this image, which was a collab between Kath and I. But you think of many of us like think of the ocean as the big ocean, and when you look at the ocean color, you say, oh wow, the ocean looks really green today, it's so beautiful. But then you think about, why is it green, or blue or purple, or whatever you see, and it's because of what's in the water. The organic matter in the water, which is all to some degree either driven by microbial processes like photosynthesis, or it's a community that's responding to that color. If there's a lot of like runoff in the ocean, then you get a set of microbes that are going to chew that up and interact with it and eat it and form sort of the base of the marine food web, which then fuels some of the more charismatic fauna that we think about in the ocean. So I'm going to tell you about how to look at the microbes, or how we look at microbes in the ocean, microbial communities to some degree. And I gave a big thanks to this content is with inspiration from a lot of different people. So thank you to all. So what I'm talking about is omics data, which is like a big broad term that sort of refers to data from biological molecules collected from anywhere, really. And so, this can be proteomics, genomics, transcriptomics. In the context of ocean samples, we're thinking about them. These samples are often collected in bulk from like a large quantity of water. And so it's not looking at individuals, it's looking at everything at the same time. And so that is where the meta is added to the omics term. So like metagenomics is sort of a metagenomic sample is sort of a collective genome DNA sample from all of the organisms collected from one particular spot. And those are often fractionated by size. So you can look at a bacterial size fraction and all of the DNA within that, or like a sort of protist or eukaryotic size fraction, which it would be bigger. Or sometimes I like to look at the viruses, which is like the smallest, everything that's less than a certain size.
Hey, Julia,
yeah,
can you zoom in on, make your text a little bigger? Is that possible?
oh yeah, I should have, yeah. How's that?
In the
That
room,
better?
is that good? It's better on my screen.
This is also on the tutorial, so you can follow along.
Yeah, that's a good point. You can make sure you sync up your hub. You can have a look at the notebook there and work along as this is going on.
Yep, and there will be some interactive coding that you can do as we move on. So then how is this data generated? It's collected in bulk from whatever size fraction you're interested in. And then, you know, there's this, and then there's a decision about which molecule you look at. And most of the sort of large omics data sets from the ocean currently are either DNA or RNA based. And so you would filter a sample, and then you're collecting all of your cells on this filter. You take that filter and you extract your molecule of interest, either DNA, RNA, or proteins is another option. And then you send that off for sequencing. And so when you do that, what you get is either a metagenome or a metatranscriptome. And so metagenome is sort of the collection of DNA that, or a representative collection of DNA that was in a sample. And a metatranscriptome is a representative collection of RNA. And so what's the difference between a metagenome and a metatranscriptome? So a metagenome is the genomic information. So it's the entire the entire potential of a cell. You know, our genomes, every cell in our body has a genome in it, but that entire genome is not necessarily being utilized by that particular cell at any given time. And so that's where a transcriptome comes into play. And so that sort of represents the RNA in a sample. And a lot of that is messenger RNA, which is RNA that's encoding genes that are likely being utilized by the organisms that that original sample came from. And so a metatranscriptome gets you a little bit closer to what's going on versus a metagenome, which is going to tell you sort of who's there and what they're capable of. And so I just went over sort of this, what can these two different data types tell you. DNA, a DNA metagenome can tell you about the presence of individuals and their what they're capable of, their potential, versus a metatranscriptome will tell you about what they're actually, what you say is transcribing, or what they're actually doing, or at least advising the cell to do. If you were to add a proteome, that's even closer to what they're actually doing. But we can talk about that later if you want. And so within these data, you can get taxonomic information. And so what I mean by that is just sort of identities. They'd say this type of bacteria or that type of bacteria, or that's an archaeal cell, or that's like an algae, or this particular type of algae. And then with function, it's like once again what they're doing. So like a photosynthesis gene would be a functional signal, or other things like that. And so these sort of go hand in hand. Like if you, for the most part, like if you get a taxonomic profile of a community and that changes over time, you can imagine that the functional potential of that community is also changing, because different individuals are capable of doing different things. But depending on your question, you're going to want to focus on one or the other. And so when you do this, when you filter your samples, extract your molecule, and then sequence it, what you get back is a lot of data. And so what does those look like? They come in the form of FASTQ files and FASTA files. And so these are sort of raw sequence files. FASTQ files are raw sequence read data that also has sort of the quality of the data incorporated into it. That's what the Q is. Versus the FASTA file is a sort of a raw sequence, is a file that's giving you just the sequence information with headers associated with each of them. And I should have given examples of what those look like, but I apologize, I didn't. But you can, if you just go to NCBI, which I'll link in a second, you can find examples of these files there. And so, okay, so now you have your data, you have your raw reads, which are literally A's, T's, C's, and G's that are indicating sort of some genomic information about the sample that you collected. And so how do you use that information to learn anything useful? One way, which is sort of what I'm going to go over today, is read profiling. So it's used to sort of access as much information as you can get out of a metagenome by sort of cataloging and categorizing each individual read within a library that you collect, a metagenome or a metatranscriptome. And so if your sequence data is going to look something like this, where you have all of these teeny little reads that you got that represent different teeny little pieces of DNA sequence that you collected from the environment, using sort of a reference, which is shown here, you can take these reads and map them to your references to figure out where those reads came from. And so it's like, this is a very sort of standard method in bioinformatics. And it all relies on a database. And so I'll talk about that in a second, but first I'll talk about the tools. And so these sort of read mapping tools, there are lots of different ways to do this, but one tool that's very fast and efficient and pretty effective is Kaiju. It's called Kaiju, and there's another one that's called Kraken 2. There's a theme there with the names of the software that are really efficient at comparing reads to a database and classifying them accordingly. And so how these databases work is that you have you have two components to the database. You have your reference genome, or a lot of reference genomes in this case. And from that reference genome, you get a collection of sort of translated protein sequences that come out of that genome. And so this is your database. And then the way that the tool works is that you start with your RNA or DNA sequence, and then you actually translate it into protein sequence, which makes it the search a little bit faster. And you compare those protein sequences to your reference proteins, which then get mapped back to your reference genomes. And from there, all of these reference genomes are classified. And so if you have a read that matches a reference protein that maps back to a genome, you can then say, okay, that read probably came from this type of organism. And these tools are really efficient because of the way that they've sort of been built. And they're great, but they're also as good as the reference database that you use to classify them. And so these classifiers rely on external databases that are then they're the external databases that then are put into a format that the tool can use. Sorry, my words are not coming right now. But so some of the databases that these tools use are the NR, which is sort of non-redundant proteins from bacteria, archaea, and viruses from the National Center for Biotechnology Information, which is an NIH body. And from there, there's also another sort of more curated collection of genomes called RefSeq that you can compare these reads to. Or another option is ProGenomes, which is which is a big database put together by ENA, or the European equivalent of NCBI, that has even more genomes associated with it, including MAGs, which I'm not sure I'm going to dive into at the moment, but or I might have to, we'll see. But so the point is that these databases are great, but one sort of issue with both of these is that most of the microbes that have been sequenced and characterized are ones that have been grown in the lab. And when you're looking out into external environments, such as the global surface ocean, those microbes are not necessarily going to match the lab-grown microbes very well. And this is sort of taken care of a little bit by, as we accumulate more and more sequence information, we get more and more genomes from environmental samples. But these genomes are not from actual individuals. They're often a sort of combined genome that's generated from a metagenome. And so it's like a representative of some community genome that existed, but not from an individual. And those are very helpful and informative, but they miss a lot of the genetic information that is included in an individual's genome that might not be shared with other individuals that are also in the environment. And so there's a lot of information that's missing, I guess is what I should say, from these databases when you're really looking at environments that haven't been studied extensively. And so for the purposes of this tutorial, I'm going to show you results from using an alternative database that that we've developed at Bigelow that uses a different type of reference genome. And so these reference genomes are called single amplified genomes, or SAGs. And so we have a technology at Bigelow where called single cell genomics, where we can take a sample of ocean water, or from anywhere. We could do soil, we could do anything, but we've done a lot of ocean microbe sequencing. We take that sample, we run it through a flow cytometer to separate the cells into an individual line, essentially, and stain them with a fluorescent stain so that we can tell that it's a cell. And then we can sort them into wells on a plate. And from there, we can extract the DNA from those individuals and amplify that DNA so that we have enough to sequence. And then we can generate individual genomes from individual cells. And at Bigelow, we've done this at scale. We have a project called Global Ocean Reference Genomes. And so we did a project, our initial iteration of this project was done on cells from the surface, tropical surface epipelate, the tropical surface ocean. And we generated 12,000 plus individual cells like this, and sort of looked at how it stacked up against other sort of ways of other reference genome collections that are out there. And what we found was that using SAGs, or these individual, a database of generated from individual genomes rather than from community genomes and cultured isolates, was way more representative of the sort of microbial diversity in samples across the global ocean, or the surface tropical global ocean in particular. And so this is one figure sort of demonstrating that. And what we're showing here is that when we mapped reads to Gorge, or this single cell database, we were able to recover, we were able to sort of classify up to 40% of the reads in that sample, which is very good for this kind of data. And by contrast, when we looked at, this is sort of all other sort of reference databases from marine microbes and how they stacked up. And so our collection of cells, bacterial cells, was much more representative and did a much better job of identifying and linking reads from metagenomic samples across the globe. And so in the map here, all of the stars indicate samples where our single cell genomes came from. And then these blue circles indicate how well the metagenomic data or the metagenomic read data that from Tara Ocean samples recruited to our SAGs. And so, you know, we only were able to sample from the Atlantic and Pacific Ocean for the most part, yet we were able to recruit really well from the Indian Ocean, which was really interesting to see that the microdiversity and the microbial genomes that we were able to sequence showed or allowed us to get a lot more information out of places that we've never been, that a lot of people aren't able to sample, or that haven't been sampled extensively, I should say. And so we found our collection is more accurate and sensitive than sort of the default databases for characterizing reads from the surface ocean, surface tropical ocean in particular. This is very biome specific. But so then these plots show, so we then took our reads and we put them into formed them into a kaiju database so that we could use kaiju in a way that that's sort of like transferable to other groups. And we looked at how it performed relative to also the sort of standard NCBI database, and it did a lot better job accurately classifying reads down to some very fine taxonomic levels. And something that we sort of implemented with this this database that's sort of different from how most of the databases with kaiju work is that we added an additional script to the workflow that allows you to actually assign functional annotations to the reads that were mapped to our database. So not only were we able to get taxonomic classifications of the reads, but also functional characterization of the reads, exactly where in our single cell genomes the reads map to and what gene that was. And so today, another great thing about a database like this is it's very finely tuned to the biome that we're interested in. And so if you want to work with, you know, if you're working with surface tropical data, you're in luck with this database. It's also much smaller, and so it runs faster than if you were to use a larger database. And so it's just a sort of more efficient and pinpointed database if this is what you're interested in. So I'm sort of going to do a bit of a demo of what the output of what it looks like to work with kaiju outputs today. And so for this tutorial, we'll use this Gorge Tropics database that I was just describing. It is a database that's publicly available. And so I have a link to the publication that sort of presents it. And there's also a repository where you can get the same, you can download the same database files that I've used for this tutorial. And so these are the specific databases files that kaiju will use to classify the reads that we're going to look into. And that's for your reference if you want to try this at home in the future. It's all set up here now for this tutorial. And I'm going to show you how this works using sort of the work kaiju as it is, but know that the group that I work with at Bigelow has implemented sort of a Nextflow workflow that uses kaiju and additional scripts to analyze a metagenomics data set and classify it using the Gorge classifier. And so it's if you want to do a full workflow and it's especially amenable to HPC environments, you should check out our GitHub repository, which you can see here. But I will also say that this is version 1, and we're currently working on version 2, which will be much more user-friendly and easier to sort of make your own databases as well. So stay tuned. And so then this Gorge classifier workflow moves beyond kaiju to assign functional annotations. But today I'm just going to use kaiju as a demo of how the database works and looks and what sort of data information you can get from it. So we're going to be running this Kaiji workflow on a collection of metagenomes from the Bermuda Atlantic time series. And we're going to use, like I said, this original version of the database. And so the Bermuda Atlantic time series, many of you I'm sure are aware of, samples at Hydrostation S fairly regularly. I think maybe monthly for the most part. And so there's a really rich data collection, and there's also some metagenomics data that's been collected through time. And so I've picked out a metadata table from a specific project that collected metagenomes monthly for the most part. And so I'm just going to sort of run you through my workflow. So I'll load all of my libraries for this whole tutorial right there. And so this is what this initial data file looks like. It's associated with an NCBI project ID, bioproject ID. And so to find this, it's referenced in the publication. And this is something you can look up on NCBI. If you put it here, oops, that's not what I wanted. Quickly do that so I can show you what it looks like. So by putting in that bioproject ID, NCBI sort of links a lot of different data types together, so you can see what publications are associated with that ID, and raw nucleotide, raw genomic data associated with the project is available here in SRA. SRA stands for Short Read Archive. And then the bioprojects itself. And so this is what we'll be looking at. So it's temporal sampling of marine metagenomes from Station Aloha and bats. So there's a lot more data than what we're going to look at today, but we'll focus today on samples from bats and from the surface. So that's where this particular table came from, is that web page. And so we've loaded it, and we're looking at, this is what this table looks like. For my interests, the columns that I'm going to look at is the run ID, because I'm looking for particular data sets. And then let's see if it shows up in this default view. No. And so the run ID is very important because that's how we're going to know how to grab our data. And then we are looking for, we're looking for collection date, cruise ID. So collection date, cruise ID, biosample, just so that we can go and look it up on NCBI if we want to get more information about a particular sample, and then depth. And so I've zeroed in on those in this particular table. And so I'm going to filter out everything except for samples from bats from 10 meters and 1 meter depth. And this table is obviously not fabulous in that it has like a, the meter, the units are in the column, but that's okay. We'll deal with what we have for now. And really, we're just interested in the surface, so we're not going to really think about the exact depth of these samples anyway. But we found a bunch of samples now from bats that were sampled at the surface. And it looks like we have almost monthly sampling throughout the course of 2 years, and then a rogue sample 5 years later. And so let's, for the purposes of this tutorial and our interest in just like taking a look at what the data looks like, let's just focus on 1 year for now. And so we'll look at 2004. And so if we filter out that, we get 11 samples that we'll look into. And that's, I mean, that's a reasonable number. We could do them all, but this will be good. And so now I've written out this particular table to a CSV, just so that I've saved just the information I'm interested in moving forward. And I've also created a list of the run IDs that I'll use to download my metagenomes. I'll run this so that my notebook doesn't break. And so here's some background things of what I did to get the data that we're going to use. So first, I downloaded these metagenomes using a package called FastQDL, which is a nicely streamlined package for interacting with NCBI and the European equivalent, ENA. And so I ran this overnight because each of these files are 1 to 3 gigabases in size. And so I did this on my own, and this is the script I used. And when I did that, what was downloaded were paired reads. So with Illumina sequencing, you actually get sort of like twice the amount of information per read that you look at. And so you get one end of so you chop, in the process of doing this, you chop your metagenome into like 300 base pair fragments or small fragments, and then you sequence both sides of that fragment. Often it will overlap. And in doing so, you actually get 2 pieces of information for each read, the forward side and the reverse side. And so those are what files we've downloaded now from NCBI. But these are huge, and we could totally run them as they are through Kaiju, but it would take a while, and then our output tables would be very big. And so once again, for the purposes of this tutorial, I've reduced these in size. Wow, this is not right. It's 100K. Each file will have 100K reads. So 100,000 reads per metagenome. So I did that using another little package called seqtk, which will help you subsample a metagenome to any level you want.
Julia,
so this is, yeah.
There's a request. Can you bump the font up a bit more, please?
Okay. That better?
I would say do it another time. In the room, is that better? I can see if you nod. Do you want it more? People in Seattle? Is that okay? That's good. Just thumbs up.
Okay, cool. Yeah, so I've got 11 pairs of metagenomic reads, and I'm going to subsample each of them, each of those pairs, down to 100,000 reads each. And so seqtk can do that for me, and it can do it randomly. But because they're paired, I want the same randomly selected sequences from both of my paired files to be the same. And so to do that, you just have to set the seed for that sort of random selection process to be the same for both pairs. And so that's what this command is doing. And so now I have my reads that I'm ready to classify. And I'm going to do that running. Oh, I thought I heard somebody. So I'm going to do that by running next. I'm going to classify these running Kaiju. I'm sorry. And so Kaiju runs in 2 steps. In the first step, the reads are mapped to the database. And then in the second step, a taxonomy is assigned to these reads based on that mapping profile. And so the first step takes a while, and then the second one is fairly fast. And I was going to demo this by running it at only 10,000 reads on one sample, but even that is too big for the Jupyter hubs. It requires too much RAM to run on the Jupyter hub, and it's not because of the number of reads that are input, it's because of the database size. And even though this database is reduced so much relative to a normal Kaiju database, it's still too big to run on this particular hub. But I was able to run it on my personal computer relatively easily, and so you should be able to too. The database itself is only 2 gigs in size. And so I'm gonna run this cell just so that I have all of these variables in memory. But these are the 2 commands you would run. So you input both your forward and your reverse FASTQ files and indicate an output file. And so, like I said, this should work on your own computer using our database, but there's just not enough RAM allocated to everyone. We'd crash it if we all ran it at the same time. But you should be able to get the results from this in the shared storage. So you can run the following cells in your own environments if you'd like to. So our output file looks like this. And so we'll read it in and look at see what it looks like. And so our output files have a couple columns. And I've sort of named the columns myself here. But so the first column sort of tells you about the status of the read. It's either U or C. U means that this particular read did not map or is unclassified. And then a C means that the read was able to be classified. This read ID is just that. It's like this output file is every single read. Contains a line for every single read in the input FASTA collection. So these tables can get really big, and if you run it on an entire metagenome rather than a subsampled one, you have to think of creative ways to sort of analyze and munge it so that you don't crash your memory. And so we have our read ID. Then our tax ID is a numeric value that is used for the second step in KJU that then assigns a taxonomy, but it maps to a taxonomy in the Kaiju database. And then the actual taxonomy is a string of names that are assigned to this, each of the reads. So first, let's just see how we did in terms of how many reads were classified. And we've got it. This is pretty good. For almost 45% of reads were classified in this particular sample. And so what does it mean for a read to be classified? It means that a read aligned to a protein from a genome that was in the database. And that genome had a taxonomic assignment, a taxonomic identity that then was able to be assigned to that particular read. And so by taxonomy, it refers to this sort of this tree of designations about who these particular what these microbes are. So we have domains, kingdoms, phylums, classes, all the way down to species. And the rank order in Kaiju is slightly different from this diagram. It starts with superkingdom, which would be bacteria, archaea, or eukarya. And then it goes down to phylum, class, order, etc. And all of these are all shown, and all of these levels are shown in one column in this output table. And in the column that I've called taxonomy. And each of these different levels is separated by a semicolon. So if you look at sort of the top 10 most common taxonomic classifications for our reads, this is what it looks like. Most of them were unassigned. But then the second most is SAR 11, which is good to see. It means this worked pretty well. So it is the most abundant bacterium in the global ocean. And so you can see these taxonomic levels going. And so bacteria, proteobacteria, et cetera, et cetera. There are NAs, and that just means that this particular read wasn't able to be classified down to those particular levels. And there are a couple of reasons why they might not be able to be classified. This happens either if the reference genome itself isn't classified to that final level, or if the read has equivalent matches from 2 different taxonomic categories. And when this happens, Kaiju assigns a classification to the read based on the last common ancestor of all of the species that that particular read mapped to. So what I mean by that is, say we have a read that maps to a protein from species A and a protein from species C, then the last common ancestor of these 2 species, if we go down this sort of family tree, is at this level, and this would be a and in this diagram, I'm depicting that as a family level classification, so it would have NAs in the other 2 spots. And so, let's sort of start to play with the results and see what they look like. So sometimes you want to just examine the distributions of how reads map to different taxonomic levels. So we'll split that taxonomy column into multiple different groups based on those levels. And so that's what this code cell does. It creates a column for each of these different taxonomic levels and the assignment to them. And so it's worth noting in these tables that we have 2 different types of null values. We have the standard NaN for unclassified reads, and we also have these NA values for reads that just weren't able to be classified down to a certain level, but they were mapped. And so that this can trip you up when working with this data. So I just want to alert you to it. But I'm going to fill the null NaNs with unmapped, just so that we're a little bit more explicit about what they are. And then you can, visualize the distribution of reads by phylum, for example. So that's what this plot does here. So you can start to get an idea of who is in this sample. And that's all well and good, but this doesn't give us a lot of context. We're like, that's nice, but how is this normal? How does this sample line up to other samples from bats, for example? And so we can check out the whole series of samples from 2004. And so all of these are in the shared Omics tutorial directory. And I've turned sort of everything that we did up here to import our table into a function. And we can read through those files and get a table that counts our read distributions by phylum. And so we look at that final data frame. What it's giving us is the library ID, a phylum ID, and then the number of reads from that library that map to that particular phylum. And now these library IDs are nice, but they represent samples from different months. And so that's obviously much better contextual information than a library ID. So we'll merge the metadata table that we looked at the beginning with this table so that we can pair library IDs with month. So now our data frame has a couple of extra columns with some more information. And from there, we can, I'm doing a lot in one line here, but I'm grouping these by month and phylum and counting the read IDs, and then creating a pivot table that can be mapped, that can be plotted. And I know I guess I have a little bit more time because we started late, but we could talk more about the code if you'd like, but I'll just, what I want to show you all is sort of what this data can look like and what it can tell you. And so here is sort of a distribution of different microbial phyla across the different months in 2004 at bats. And this is this informative? I guess maybe to a trained eye. Something that I see, well, first of all, there's way too many colors relative to the colors that we see here. And so what you can do is what's often done is you select the, most represented taxa and only label those and call everything else other. And so that's what these code cells are doing, and then replotting. So now we have a much more manageable color scheme, although there are still repeat colors in here, I'm noticing now. And a lot is up here in this sort of like very small representation groups. Anyway, so maybe this is helpful, but something that I see as a bit of a problem here is that the unmapped reads are really variable across samples. And reads can be unmapped for a number of different reasons. They could just be complete gibberish, like they could be artifacts, or they could map to regions of the genomes that aren't represented in the database. And so it's fairly common and acceptable to ignore those unmapped reads and sort of make the assumption that that the unmapped reads are unmapped in a representative way to the classifications that we do see. And so we can throw out those reads, or at least we will for the purposes of this tutorial, and really look at the results. And now this looks a little bit more, it's still maybe not super informative to an untrained eye, but you can start to see some patterns in the distributions of these different phyla over time. And I was sort of playing with this unmapped problem when I was creating this notebook. And so another thing I do in this cell is just sort of look at how the distributions of that proteobacterial group, which is the most represented in this data, how their distribution in, the shape of their distribution changes when you include or exclude those unmapped reads. And so you can see that they're not, the shapes are similar, but they're not the same. And certainly in terms of, the relative abundance of these different groups, that actual value is very different depending on whether or not you use those unmapped reads. But I would say omit those unmapped reads and just consider the ones that were mapped. And so another way to look at these data, which is maybe a little bit more, I don't know, I find it more satisfying, is to look at beta diversity or compare the structure of the population of the different populations in different samples. And one way to do that is using dimensionality reduction methods such as PCA. And this will look at sort of the overall relationships in community structure between samples. And so the next couple cells are going to set up a PCA plot. And so this time, instead of considering the phylum level classifications, we're just going to use the taxonomy column, which is sort of the entire classification for each of the different reads. And so this is creating a data frame very similar to the last version. But it's going to take a little bit of time because we're going through all 11 metagenomes again. And so now you can see instead of just a phylum designation here, it's counting all of the different reads and all of the different samples that fell to each of these different taxonomic full lineages. And so once again, I'm going to add the metadata to get my my month column into the index for the pivot table. And so now, and I've also transformed this to be relative abundance. So the percent of reads per sample that mapped to each of these different designations is what this table is showing. is it percent or just proportion? I think it's proportion. Yeah. Yeah, the proportion of rates that map to each of these different categories. And so to do PCA, it's important that you normalize your data. And this is something that Kath and I have talked about a lot. What I'll do here is a center log ratio transformation. And I'm doing it just using standard library, well, not standard, NumPy libraries, but there's also packages that have these functions embedded in them that you can use. But so there's our transformation. And then we'll plot PCA and see what it looks like. Plot the PCA and see what it looks like. So that's what this cell does. And now you can see sort of a different kind of result than before. And this, I found this output to be very satisfying. So we're labeling these points by month. And so it's sort of each of these axes are principal components that encompass some of the variance in the data overall. And so this first axis is representing 66.2% of the overall variance in the data set. And then the second axis represents a smaller proportion of it. But this is a pretty big portion of the overall variance. And sort of what I'm seeing is a story here of we have sort of the winter to spring bloom samples. And then at some point, the water column becomes stratified. And we have sort of a mixed layer that looks relatively similar to each other for the summer months. And then as winter approaches, everything shifts again in a different direction. And it'd be interesting to add another year to this to see if it goes in a circle or if it goes in some other, the community goes in some other direction for the following year. But anyway, but this is just, a start. From here, there are methods that you can use to then pull out, okay, what taxa were most dynamic over the course of these samples, and start to understand what components of your data are driving these patterns. But I don't have that in this tutorial. This is where this tutorial ends, but I hope there's probably plenty of time for questions. And I just have one more thing to say. Yeah, happy to answer questions.
Thanks, Julia. You let the cat out of the bag. I don't think anyone knew.
Oh, sorry, Kath.
It's okay. It's okay. Does anyone have any questions that they want to ask Julia? I'm not seeing much on the Slack. I personally have a couple of questions which I can throw out there, but has anyone else got anything they want to ask? I'm not seeing anything.
Oh, one more comment before I meant to say this. So I'm like a Python junkie. And so I tend to like dive into things with Python. But there are a ton of really great R packages that deal with this type of data. So if I were to do this tutorial again, I'd probably write it in R. But I was just so you're aware. I think I linked to a couple of them earlier in this tutorial, so check it out. But anyway.
And on that, earlier in the week, I mentioned that example of a project I was working on with someone where we did all our data munging in Python, and then we switched to R for some analysis and visualization. That was a project that Julie and I were working on together. And so, yeah, sometimes using both languages is good. And so I have like a bunch of questions related to, and I think I've asked you these in the past, but I'm just asking them again in this space, related to like all your different databases like you presented at the start, and the Gorge database, and how different your results are, like when you use different databases, or like, do you get multiple options? Like how does your community kind of deal with that aspect of of some of the work that you're doing?
I haven't, aside from sort of proof of concept showing that our particular database is really good for what we do, I haven't tested, I haven't really done some ground testing on like how different the results would be if you used a different database. My suspicion would be that you would probably see similar patterns, but you wouldn't be getting nearly as much information out of it. Like much fewer reads overall would be classified. And so if you're still gonna throw out everything that's unclassified and look at just what is classified, you might get similar results, particularly because, some of the dominant taxa are so dominant that they're probably driving these patterns. But you're definitely missing a lot of nuance by choosing a more generic database.
Great, thanks. There is a question now from Ben. How much work does it take to create your reference database? Minutes, months, years, something else?
How about dollar signs? I mean, but so the single cell genomes database is like a mammoth effort. In terms of the sequencing, like the prep it took to get those cells, get them sequenced, and then quality controlled. That's something I didn't cover, but like our genomes are they're high quality in terms of we've looked at them for contamination. We've only included the highest quality, most reliable genomes in this database. And so all in all, I think the generation of that particular database probably took 5 years and 3 people's time.
Yeah. There's another question. How do you publish if you have used your own curated reference database? How do you publish it? Do you publish it?
I would publish it. Yeah. Our genomes each of the genomes that are in the database are available through NCBI. But in the reference database itself, in the format that it's in, we've shared through OSF, open, I can't remember what it stands for. Maybe somebody else remembers. But anyway, it's just sort of it's a DOI controlled open database that you can submit data alongside publications too. And so that's where we have it available right now. We're thinking about, because we're working on a gorge dark where we have we're currently collecting and quality controlling a lot of cells from the dark ocean. And we'll probably assemble more and more collections of these over time. And so eventually we'll have our own public portal to do it. But if you generate your own database, I would publish those files with your paper in something like Zenodo or OSF or something like that.
Great. Someone shared the OSF link on the Slack, so thank you.
Thank you.
Any other questions? If not, I will say thank you, Julia, for that. That was great. I really enjoyed it. I've seen you give many science presentations, but it was fun to see some of the code behind it, which I don't normally get to see. So thank you. And I think we're scheduled for a break, but Woo Jung, did you want to say something?